Next, let us learn about the application of biotechnology in the field of medicine. Recombinant DNA technology helps for mass production of safe and more effective therapeutic drugs. It helps in the production of safe therapeutic drugs. The products from non-human sources induces unwanted immunological responses. But recombinant therapeutics does not have any such problems. So the benefits are, first, it is very safe. Second, does not create unwanted immunological responses which is created when these products are obtained from non-human sources. And at present, about 30 recombinant therapeutic drugs have been approved. Of these, 12 are marketed in India. The different types or applications in medicine are genetically engineered insulin, gene therapy, and molecular diagnosis. We know what is insulin. Insulin is a hormone which is secreted by the pancreas. And what is the function of insulin? It helps to control, or the deficiency of insulin causes diabetes. So insulin is having a very major role in controlling diabetes. So let's learn about genetically engineered insulin. Insulin is used to manage adult onset diabetes. And insulin from the pancreas of animals like cattle and pigs causes allergy and other types of reactions to the foreign protein. Okay, so when these insulin was isolated from non-human sources like cattle and pigs, what happens, as I told you earlier, it creates immunological reactions, allergic reactions. Okay, so now it is possible to produce human insulin using the prokaryote, that is bacteria. The two short polypeptide chains are chain A and chain B. These are the two polypeptide chains, chain A and chain B, and they are linked together. They are linked together by means of disulfide bridges. So the two chains, polypeptide chains, chain A and B, are connected together by disulfide bridges. In mammals, insulin is synthesized as a prohormone, as an inactive hormone. And this prohormone needs processing to become a mature and functional hormone. This is a prohormone. What do you see? This is the chain A and chain B. And in addition to it, there is a C peptide. It has an extra stretch called as a C peptide. And the C peptide has to be removed so as for the prohormone to become an active hormone. In 1983, a company, an American company called as Eli Lilly Company, they prepared two DNA sequences. The DNA sequence corresponding to chain A and chain B of the human insulin. So they synthesized, or they made a sequence, two sequences, sequence A which codes for A polypeptide and sequence B which codes for the B polypeptide. And they were introduced in the plasmids of E. coli to produce the insulin chains. Chains A and B were combined by creating disulfide bonds to form a human insulin. So they were separate. These two DNA sequences were separately introduced into the plasmids of E. coli. We know plasmids are autonomously replicating circular DNA. So they undergo replication, and when these are cultured or cultured in large quantities, the transformant starts to produce a particular gene product, that is the chain A, in those plasmids which have the sequence A. And those plasmids who are having the sequence B will start to produce the polypeptide chain B. And they are extracted separately. They are extracted separately and they are purified and combined together by disulfide bonds to form human insulin. Next is gene therapy. It is a method to correct a gene, a defective gene, either in a child or an embryo. A child or an embryo who's having a defective gene is corrected. Here, genes are inserted into a person's cells and tissues to treat a hereditary disease. We know when a particular defect is present in the gene, it is a hereditary. It can be transferred from the parent to the offspring. So these hereditary diseases are controlled by the corrective genes which are inserted into the person's cells and tissues. And it compensates for the non-functional gene. A defective gene is non-functional. So when the defective gene is removed and a corrected gene is inserted, the non-functional gene is compensated. The first clinical gene therapy was done in the year 1990 and was given to a 4-year-old girl who had ADA deficiency, that is adenosine deaminase deficiency. So the gene, particular gene was defective. So they were in that particular child, that gene was not able to express and produce the enzyme called as adenosine deaminase. This is a girl who was treated for ADA deficiency and is caused due to the deletion of a gene. The deletion of a gene, the gene which codes for adenosine deaminase, was deleted. And what is the importance of this enzyme? It plays a very important role in the immune functioning of the body. And treatment by bone marrow transplantation or enzyme replacement therapy is not completely curative. So these are the first two methods for treating ADA deficiency. That is, first is bone marrow transplantation and second is enzyme replacement therapy, that is injection of ADA, repeated infusions of ADA enzyme into the body. Those two methods were not completely curative. The curative method is the application of biotechnology, that is gene therapy. So these are the three methods of correcting. First is bone marrow transplantation, second is enzyme replacement therapy, and third is gene therapy. Collect lymphocytes from the patient's blood and it is grown in a culture. So the patient's lymphocytes are collected and they are grown in a culture and they introduce a functional ADA-cDNA. So the functional gene is the ADA-cDNA into the lymphocytes. And what is a vector which helps the introduction of the gene into an animal cell? It is a retrovirus. So using a retrovirus as a vector, the specific gene which codes for the enzyme adenosine deaminase, that is the gene is ADA-cDNA, is introduced into the lymphocyte using a retrovirus. This should be periodically repeated as lymphocytes are not immortal. It has a very short lifespan. In order to have a cure, what happens? These lymphocytes, as they are immortal, this process has to be repeated periodically. That means it has to be removed from the patient, and again using a retrovirus, the gene has to be introduced into the lymphocyte. In order to overcome this limitation of repeated periodical replacement of these lymphocytes, these modified lymphocytes, if the ADA gene it's taken from the bone marrow and it is introduced into the cells at the embryonic stages, it could be a permanent cure. So that is a permanent cure for gene therapy for adenosine deaminase deficiency. Thank you.